Yeah, my name is Scott Tebbutt. I'm a PhD basic scientist working at the University of British Columbia, and I'm Chief Scientific Officer for the Proof Center of Excellence. So, a blood biomarker is effectively, in our context at least, it's a molecule. Right? And molecules are usually produced by the action of genes, targeting, producing proteins that could be enzymes. And then the enzymes will clip something and turn one molecule into another molecule, and often these can be metabolites, or you could actually be looking at the output of the gene itself, either RNA, the messenger RNA, or you could be looking downstream from the messenger RNA, and that could just be the protein. Right? So these are basically different molecules that you can measure in a blood sample, and the idea, if they make a good biomarker, is whether they can actually be used. If you measure some of these molecules in one type of people, right, one person, compared to another person, and if there's more of them in this person who has heart failure, and there's less of them in this person that doesn't have heart failure, then effectively they're a biomarker. So the central dogma, if you like, of sort of molecular biology is that we have our inherited DNA that we inherit from mums and dads, right? And that genetic material sits in all of our cells, but the outputs of that genetic material, the gene expression is different from one cell to another. So a brain cell will have a different set of genes expressing compared to a liver cell, right? And in the blood, we have different types of blood cells, immune cells, if you like, and they'll have different patterns of gene expression depending on what the cell does, right? What its function is in the body. And the idea is if you have a sort of a condition, a health-related condition, that these cells will change their behavior somewhat, right? And so their gene expression outputs will change. And the idea is that you have to measure these outputs at the molecular level, either at the RNA level or the protein level, or downstream, the metabolite level. And if you can find molecules that do differentiate these two patient groups, then you have a biomarker. But it has to be reproducible, so you have to do enormous numbers of studies to show it's true, because we're all different, right? Even though our DNA is 99.9% identical, we are still all very different, and therefore you don't want to be getting biomarkers for the difference between men and women. You don't want to have biomarkers for the difference between, a 20-year-old and a 30-year-old. You want to have a biomarker that can tell the difference between someone with impending heart failure compared to someone who doesn't have impending heart failure. So those are the, and it takes a lot of work and effort to actually be able to really discriminate between these types of things.